So you saw some of this pathology in the last presentation too. So you all know that Alzheimer's disease is associated with these extracellular blobs of a protein called A beta, the amyloid plaques, and also intracellular inclusions of a different protein called tau that form these tangles inside neurons. And at some point, somehow, this amyloid and tau cause neurons to die in the brain that physically results in a shrinkage of the entire brain, so that late in the disease, you have a loss, particularly in the temporal lobe, here in the hippocampus and temporal lobe, which is responsible for the short-term memory. And you also have a lot of loss in the frontal lobes of the brain too, which control what biologists call executive function, but as doing the right thing, doing it in the right order, and at the right time. So, back in the dark ages of, 1999, we had to diagnose Alzheimer's disease by looking at a brain specimen that was collected at autopsy. Most people don't want to do that. So the field in general has started to come up with these fancy scans to look at Alzheimer's disease. And the first, so these are PET scans that are giant machines that look like an MRI. You use a particular drug that travels through the bloodstream and binds to the pathology in the brain. So it's sort of like an MRI with a contrast agent. And the image doesn't come out as a brightly colored thing like this. Usually it comes out as a black and white image that the computer then assigns colors to. And so by convention, the field has decided that they're going to put these cold colors like blue and green, meaning there's not much there. And then they're going to use the bright colors like red and yellow to show where there's a lot of binding. And in a patient that has Alzheimer's disease, you can see a ton of amyloid that's present in the frontal lobes and also some in the temporal lobe as well. More recently, maybe 10 years ago, we started developing molecules that allow us to label tau. And so you can start to see behind the little thing here, there's tau in the Alzheimer brain that's localized in the temporal lobe compared to the person that doesn't have any tau in their brain, doesn't show any of this bright color. And you can also see how much improvement in the imaging abilities we've had over the last 10 years, where here you got a blob of something that doesn't really look like much compared to a real brain in the more recent pictures. So now that we have these scanning things available, scientists have started looking at what is the time course of pathology development in Alzheimer's disease. And it turns out that the first thing that happens is you get amyloid in your brain. And this is part of the evidence that suggests that amyloid is what starts the whole process. It's not the end, it doesn't do everything, but amyloid is a key driver of the pathology. And over time, if you look at how much pathology you get, come on, pointer, going that way, you can see that over time you get more and more and more and more amyloid until finally you get all the amyloid you're ever going to have. You don't get any more after that. But some point later in this green line, you start to develop tau, right? So there's a gap between when tau starts to form, and then once the tau starts to form, there's a further gap between the time that the brain starts to degenerate. You start to have memory impairments that are bad enough that you're going to go see a clinician about what to do about that, and ultimately develop symptoms that interfere with activities of daily living later in the disease. And by looking at many, many scans over a large number of people, it now looks like we have a preclinical phase of 10 to 15 years, a decade of time, when people are building up amyloid in their brain before they get to the stage when they have memory impairments that impair their quality of life. So this is optimistic. We have a long time to do something If we can start using these scans more to identify people that are beginning to have amyloid, we can do something about the amyloid, and hopefully that will take care of all the downstream problems here. We don't know for sure if that will take care of all the problems, but there's something, amyloid is causing something to lead to all of these effects. And it also points out the fact that Alzheimer's disease is considered a secondary tauopathy, right? So the amyloid comes first and then the tau comes first, or comes second. There are different diseases that are primary tauopathies, where the whole problem is caused by tau and don't have amyloid. And those are diseases like frontotemporal dementia. And there are some others too that are primary tauopathies that look very much the same, cause dementia. The symptoms are a little bit different. So we wanted to model this time course in animals. And so the way that most people have made mouse models of the disease is they genetically manipulate the animals so that they have mutations that are associated with disease, and they express those mutations from the time they're conceived. They start having amyloid, basically from the minute they're born. And that's not what actually happens in Alzheimer's disease. And so we set out to develop a model where we could mimic this time course of amyloid and tau pathology. And so many, many long years ago, we developed these kind of mice that have mutations in the amyloid precursor protein and the presenilin gene that cause Alzheimer's disease in people, right? If an individual is unlucky enough to get at least one mutation from one parent, they will definitely get Alzheimer's disease. And those are early onset cases that are extremely rare in the population. Most people do not have those mutations. But we started with these mice and we generated mice that have these amyloid plaques. And at the time, 25 years ago, this was a big deal and it got us a cover of a magazine. So there's my great picture of an amyloid plaque in a mouse. And of course, we can compare those to the control mice that don't have any amyloid. And because Alzheimer's disease is huge in women and also men get it, in all of our experiments, we want to use both male and female animals so that we can compare what's going on in the two. Here is a little picture of a mouse brain. You see it kind of looks different than a human brain, but all those little tiny black dots that you can barely see is an amyloid plaque in the mouse model. So we waited until the animals develop a lot of amyloid pathology according to that time course. And then we use a gene therapy approach to try to force them to make tau. And we are also using the genetic sequence that was discovered in humans that cause Alzheimer's disease. So it's not like we put a human gene in there, but we use the sequencing information to construct an artificial gene, right, that has the same sequence as a human gene. And so we put the tau into the animals that, oops, that was too fast. We put the tau into the animals that already have the amyloid or don't, and then we wait a period of time for them to develop their tau pathology as well. How do you look at learning and memory in a mouse? Well, we make them go in mazes. And the one that I developed was, it's a cute little swimming maze. So, like most mammals, they know how to swim. You throw them in the water and you teach them, you train them to swim to an escape platform to climb out of the maze to get their little reward. So we can put the mice in these and they can learn to navigate this maze, and that's how we test their memory. We can test their short-term memory, we can also test longer-term memory. And then we take out their brain and we cut it up into itty-bitty little pieces, and we do something different with each of the little itty-bitty pieces. We do biochemistry on some of it, we do anatomy on some of it, we take out the RNA and we measure the expression of the RNAs that way. We do work with DNA as well. So we take out little bits of the brain and we do all kinds of stuff with it. So here's what happens in the mice if you do that. So this is a brain section from a mouse that's stained for those tau tangles. Here is an amyloid plaque stained in pink, and you can see all of these tau tangles that form in the animal that had the amyloid pathology to begin with. But the animals that don't have amyloid to begin with, they hardly make any tau at all. So there's something, you can get tauopathy formed if there's no amyloid, but when you have amyloid, it's much, much worse. And so we think this is going to be a really good model to look at what happens. Can we slow down the rate of progression between an amyloid positive situation and an amyloid and tau positive situation? That's our goal with this. And when we take a whole bunch of sections, so one grant reviewer said to me, well, a picture is worth 1,000 words, but a number is worth 1,000 pictures. So we have to quantify all of this histology that we laboriously do. And when we measure the amount of how much this dark staining is present on a bunch of sections from a bunch of different animals, each one of these little dots is one mouse, then we find out that throughout the brain, especially in the regions that are important for Alzheimer's disease, we have more tau that develops in the presence of amyloid. So now I want to back up a little bit into this genetics business, right? So now we have sequenced all of the genes from a few people. And so we're starting to do that more and more. We're starting to get into the what's been called personalized medicine, where we're starting to look at the individual genes. And this is what we know about genes that cause Alzheimer's disease. So I mentioned these genes up here that cause Alzheimer's disease. So if you have these genes, you have an extremely high risk of getting Alzheimer's disease, but they're exceptionally rare, right? Almost nobody has them. It's very, very rare. They run in families. The families are, it's probable that all of those families have been identified now, unless they're in some strange exotic location. But now, by pooling together the genetic sequencing that's been done by all the Alzheimer researchers across the world, we're starting to identify other genes that increase risk for Alzheimer's disease. And one you might have heard of is the biggest one, apolipoprotein E variant 4. So it's the apolipoprotein, or ApoE4. And that causes, if you have one copy from one parent, about a 3-fold risk in Alzheimer's disease. And if you have 2 copies from both parents, it's about a 10-fold increased risk of developing Alzheimer's disease. But now we're finding all of these genes over here that are really common, right? Half the people have some of these mutations, or they're not mutations, they're variants of genes, because they're very, very common. But they increase risk a tiny, tiny little bit. So maybe a 5% or a 10% increase of risk. Note that different people might have 1 or 2 or 4 of those, variant alleles that change risk of developing Alzheimer's disease. And when we start looking at these genes, about half of them have something to do with immune function. And here is somewhere in here, I don't know, maybe CD33 is in there, one of those. That one, yes. It's that one right there. So it increases risk of Alzheimer's disease a little bit in people. And so these blues are colored differently for some other reason, but it turns out that a lot of these genes are controlling immune function. And so the genetics are telling us that we have to pay attention to the immune system in the brain. Something is going on with them. And so in the brain, the immune system components are these microglial cells. And this is a picture of a very blown up microglial cell, colored artificially green. They're not really green in real life. But you can see that it's a very small cell, which is called micro, and it has these very beautiful and elaborate processes that are all spread out and covering a whole lot of things. They're evenly spaced throughout the brain. When the amyloid plaques come around, again stained with this pink stain, the microglia are trying to cope with all of this amyloid. And so they go near it, they surround it, they become activated, they try to phagocytize or take in, they eat, they engulf the amyloid. And if you have really good eyes, you might see a couple of these that look like it might be one cell with amyloid inside of it. Other people have maybe better pictures of that. But these are the microglia that are trying to respond to the amyloid plaques by phagocytizing it. How come they don't? That's a dilemma. We're wondering, why can't these microglia phagocytize all the A-beta and get rid of it and cure the problem? Why shouldn't they do that? It turns out that people get Alzheimer's disease when they're old. They've already done their reproductive duties and contributed to more people in the world. And so elevation or changes in gene expression can only happen to things that take place in people before they pass on their genes to the next generation. And so if there's evolution out there, there's probably not a lot of evolutionary pressure for the microglia to do something specific to the amyloid, because the microglia didn't ever see that. And so the microglia are just trying to do something that they already know how to do, some program that they have during development, et cetera, that is reacting to this amyloid and is capable of doing something. Okay, take a step back. If we don't talk about genetics, what's the biggest risk factor for Alzheimer's disease? Well, it's too many birthdays. When you get, Alzheimer's doesn't happen in young people. It has to occur in the aging brain. And so my whole lab is studying what is it about the aging brain that causes these changes that allow the amyloid and the tau pathology to be bad. I guess I didn't talk about resilience, but okay. And this graph here shows you that as you go older in age, the amount of people that get Alzheimer's disease is exponentially increasing. In both women are here and men are here. It's looking like there might be a tailing off later in life. So people that live to be 100 might be having a lot of these protective alleles that, let me go back to that. Oops, that's the wrong way. So I forgot to tell you about too, these are all the genes that cause increased risk. But recently we've been finding newer genes in the last year that are protective against Alzheimer's disease. And my lab is studying very hard some of these protective genes that are going to slow the rate of Alzheimer's disease, and we're trying to understand how they affect microglial function as well. And some of these protective genes are in the some of these protective genes are in the ApoE4 locus, but there are also newer ones that you heard about earlier, and another one in a protein called realin that we are working very hard on in other projects in the lab. So one of the things that happens to our cells as we age, as you all know, there's a lot of wear and tear that accompanies the aging process. And so our cells become aged as well. And you might use the word senescent as a synonym for really old, but in biology, it means a very specific thing. Cells that become senescent, they stop dividing, they don't have any more proliferation. That's here on this cycle here. So if you're a skin cell and you cut yourself, the skin cells proliferate and repopulate and cure heal the disease. In your brain, the microglia and the astrocytes can proliferate, but the neurons cannot. So anyway, this is one of the effects of senescence. And the senescent cells, they change their shape. They start making new things that they shouldn't make before. They secrete a lot of factors, so they release chemicals that signal nearby cells. I'm senescent, why don't you join me and become senescent too? And so some people have called them zombie cells because they're not dead, and they conscript other cells near them to also become senescent. And there's a lot of ideas that these senescent cells might lose the normal functions that microglia have that are beneficial for removing the amyloid, or they may gain new functions that are detrimental and contribute to how amyloid induces tauopathy in these mouse models. So when scientists design a series of experiments, it's always good to do some experiments where you increase your whatever it is, the gene, the protein, the senescent cells, whatever, you want to increase them to see what happens, do a proof of concept type experiment to see where that leads you. But we also, in Alzheimer's disease, are very interested in coming up with some therapeutic strategies to reduce the area that we're interested in. And so in two aims in this project, I'm going to try to promote microglial senescence with the hypothesis that that is going to make the cognitive impairments worse, the neurodegeneration worse, and increase the amount of tau that the brain has. And on the other side, we want to get rid of these senescent microglia because we hope that that will reduce the amount of neurodegeneration, cognitive impairment, and pathology development. And so just, to give you a little details about how these experiments are going to work, in aim one of my project, we're going to start with these mice that we have. We're going to grow them until they get to an age where they have a lot of amyloid. We're going to use our gene therapy approach to put in the tau that's going to form the tau. And then the little trick we're going to play is we're going to give them a chemical that causes the death, really, the removal of the microglial cells in the brain, and with them the senescent microglia as well. And then we're going to wait. We're going to do our behavior. We're going to do our biochemistry. We're going to do our histology, all of the measures that we do down here, with the hypothesis that this is going to be a beneficial effect. And as a proof of concept study, we also want to do the reverse. We want to make them more senescent cells. And this is the little creative idea that I have. So in normal aging, cells can divide for a finite number of times, and then they stop dividing and they go into the senescent state. So I'm going to take these microglia that are in these mice here, and I'm going to treat them with this chemical that kills the microglia. But then I'm going to take away the drug. So then, obviously nothing is perfect, and so it's not going to kill all of them. There's a few that are left behind. And those few that are left behind are going to proliferate to repopulate the brain. And then I want to do that. This is supposed to be a little pile of mousy food. It doesn't really look like that, but nonetheless, that's what it's supposed to be. We're going to give this drug through the chow of the animal. So just like you would feed your pet, you hide the drug in the food because that's the only way they'll take it. And so we're going to do this. We have these cycles of this drug that are going to force the microglia to enter senescence. And then we're going to put the tau in there. Again, the same thing at the same amount of time. We're going to measure how much learning and memory deficits they have, how much neural atrophy they have, what happens to their microglia, do they become senescent, all of those things. And of course, we have have a little bit of information that this is a feasible approach. So this is a microscope slide of the microglia are artificially colored with this green color. And in the control condition, you can see that there are these green cells. You might be able to see that they have little processes coming off them. But then when we give this drug to deplete the microglia, you don't see any more of these little green dots over here. And this one is reduced in size and complexity of its processes. And when we don't count, we have the computer count. We see that this is the time course with respect to how the microglia are lost. So we're looking over 2 weeks of time here. This drug kills the microglial cells. When we take away the drug, the microglia do come back, and I elected not to show you that. But after we do this 6 times, or well, we did the drug treatment 3 times or 6 times, and then we give them time for the microglia to repopulate. And these graphs down here show you that we are getting expression of cellular senescence in these microglial cells. And so these are just graphs of certain proteins that microglial cells, senescent cells have a shorter telomere. That's just a characteristic that they have, and we show that these cells also have that. And then this is a microglial cell. This is the nucleus of the cell, and we know it's a microglia because it has this green staining around it. And then it has the marker for cellular senescence in these little red dots on it too. So we know that this is a picture of a senescent microglia that we made senescent by doing this drug regimen. And so that's the experiment that I'm going to do with the grant fund. Well, actually, these are the people that are going to do the work. And I'm going to just tell them what to do. These are former students in the lab. We always, we don't do science in a silo, and so we have collaborators that help us with various aspects of things. I do get funding from the National Institute on Aging to look at aging effects and Alzheimer's disease. I have received grants in the past from the Alzheimer's Association, and I'm really excited now to be able to add a new logo to my acknowledgement slide. So I want to say thank you very much for everything that you do to promote research and science. Thank you for the award to myself, and thank you for all you do. I'm really very appreciative of your efforts. Thank you so much.